Okay, good morning everyone. Today we are going to have a lecture about blood vessels and hemodynamics. Our today's plan is structure and function of the blood vessel, hemodynamics principles and their physiological interpretation, capillary exchange and its mechanism, blood pressure and the factors affecting on it, and control of blood pressure and blood flow. First of all, let us start to discuss about what type of blood vessels we know. We know five types of blood vessels. It is arteries, which carry blood away from the heart. It is arterioles, capillaries, venules, and veins. All of these vessels consist generally of the three layers or tunics: tunica interna or intima, tunica media, which mainly will be presented by the muscle layer of smooth muscles, and tunica externa, which is going to be presented by the elastic fibers or collagen fibers. Modification account for five types of blood vessels and their structural functional differences. So it means that the arteries to some extent will be different from the veins in their structure, and about what we are going to discuss a little bit later. So let us firstly stop on the structure of the tunica intima. Its inner layer of the blood vessels is directed to and contact with the blood. It contains endothelium, which continues with endocardial lining of the heart, and also it plays active role in vessel-related activities. Tunica media is mainly will be presented by muscle layer and connective tissue layer. Muscle layer will be presented of the smooth muscle. It has greatest variation among vessel types. For instance, in arteries, it will be more developed in comparison with veins. Smooth muscles will regulate diameter of the lumen. It means smooth muscles will be responsible for constriction, which is called vasoconstriction, and lead to decrease the lumen, or vasodilatation, which is responsible to increase diameter of the lumen. Tunica externa mainly presented by the elastic and collagen fibers. In a different type of vessels, it will be different. Also, it helps anchor vessels to surrounding tissues. In this slide, you could see the structure of the tunica vessels. So you could identify that this tunica interna, which mainly consists by the endothelium cells, tunica media, which presented by the layer of the smooth muscles, and tunica externa, which will cover the vessel, and it will be presented by the collagen fibers or elastic fibers. Okay. So now let us talk in detail about the arteries. Arteries contain three layers as a typical blood vessels. They have very thick muscular layers, that is why they're responsible for vasoconstriction and the vasodilatation. Due to their high compliance, vessels wall are able to stretch or expand in responding to the pressure without tearing, and and as a result, as I mentioned previously, it will lead to the vasoconstriction, which will lead to decrease of the lumen diameter, or vasodilatation effect, which is going to lead to the increase of the lumen diameter. Elastic arteries, the example of the elastic arteries, it will be the aorta. It is mainly the largest arteries which with the largest diameter, but walls relatively will be thin. They have a very high developed the external tunica, which is presented by elastic fibers, functions as a pressure reservoir, and they help to pull blood from the ventricles toward to the arteries. Also, they are known as a conductive. Also known as conducting arteries, they conduct blood to the medium-sized arteries. Now let us saw blood will flow from the aorta to the medium-sized arteries. which have very developed muscle layer, but at the same time they have a fewer elastic fibers than in comparison with the aorta. Walls, they are relatively thick. They are capable to create vasoconstriction and vasodilatation, and also this type of arteries are called distributary arteries. This type of arteries to some extent are responsible for anastomosis. It is a union of the branches of two or more arteries supplying the same body region. This will provide alternate routes which is called collateral circulation. Arterioles. This is the smallest part of the blood vessels in the artery system. They serve as a kind of tap for microcirculation system. This tap to some extent could open, so it will lead to the vasodilatation, and sometimes they could close, it will lead to the vasoconstriction. In this case, they will regulate the blood flow to the specific region and the blood pressure in that region. Metarteriole has a precapillary sphincter which monitor blood flow into the capillaries. Sympathetic innervation and local chemical mediators can differently affect on their diameter. So mainly the diameter of the arterioles will be controlled by the three mechanisms. The first mechanism will be the local control, which will be done by the accumulation of the vasoactive substances in the extracellular place, like carbon dioxide, hydrogen ions. Sometimes, and this will lead to the vasodilatation of the arterioles. Sometimes, due to the prolonged occlusion, after the prolonged occlusion, we will receive condition of the vasodilatation of the arterioles. So it's due to the mechanical factors, or due to some kind of inflammation damages, which will lead to increased level of the histamine, which will launch kallikrein system. They are responsible the same for the dilatation effect in the arterioles. Besides this, there is a present such substances like endothelin-1 and thromboxane, which are produced by the endothelium of the arterioles, and they're responsible for their vasoconstriction. Another two mechanism for regulation of the arterioles diameter, it is neuronal regulation, which is presented by sympathetic division. We know that sympathetic division is responsible for vasoconstriction of vessels. Also, some part of sympathetic division which produces nitric oxide is responsible for its vasodilatative effect. And another part mechanism which is responsible to regulate diameter of the arterioles, it is hormonal regulation. It means hormones are responsible to change the diameter of the arterioles, like angiotensin II, vasopressin, epinephrine. This is a very important and they have mainly vasoconstrictive effect on the arterioles. Arterioles, another name for them, it is resistance vessels, which are responsible for resistance. Resistance is opposition to the blood flow. And due to the increase of resistance of this vessel, in this situation will rise blood pressure. Capillaries in the vascular system are responsible for making such specific capillary network which will surround organs, and they will supply the tissues by the metabolic products, by the oxygen, and so mainly in the capillary network will place the process of exchange. As this is the smallest blood vessels that connect arterial outflow with the venous return. Microcirculation flow from metarteriole through the capillaries and into the postcapillary venals. This type of vessel are named exchange vessel, as the primary function of them is to exchange of the metabolic products and also gases. That is why they have a lac tunica media and lac tunica externa. Mainly they will contain the layer of the endothelium cells and the basement membrane. Sometimes they also will be present the specific windows or pores through which will water and low molecules with a low mass could exchange without any problems. Capillary beds, they will arise from the single meta-arterioles, and while the motions which is present here, it's intermittent in contraction and relaxation of these capillaries, and through 4 channels bypass capillary bed. So in this picture, we could see the structure of this capillary bed, which consists of the meta-arteriole, which have present pre-capillary sphincters, which could relax and sometimes constrict. Then there is a capillary network. After that, blood will flow through the post-capillary venule, and it will flow toward to the venules and veins in the and directly to the heart. Then there is 3 types of the capillaries. The first type, it is a continuous type of capillaries in which endothelial cell membranes will form such continuous tube. Fenestrate, they have a fenestration of pores. You could see here such fenestration through which will happen processes of the exchanges. And sinusoid capillaries, if we compared it with fenestrate, you could see that these pores will have a bigger size, bigger diameter, and that is why they will they will have such unusual large fenestrate. So process of exchanges in such type of capillaries will be better in comparison with the previous 2. So after the capillary network, the blood will flow to the venous system. So veins, first of all, let us discuss the general structure there. They to some extent are different to the structure of the arteries. In general, they will have very thin walls in relation to the total diameter. Tunica interna will be thinner than in the arteries. Tunica media will be presented with a little smooth level of with a small level of the smooth muscles, and tunica externa will be the thickest layer in comparison, even in comparison with the arteries. Also, what is specific for the veins, it is the presence of the valves, which is responsible to prevent the backward blood flow to the capillaries, and as a result, blood will flow directly to the heart. This mostly, the main function of this valves is presented in the limb veins due to the muscle contraction, and they were responsible to pump blood toward to the heart, and as a result to return back into the heart. So muscle contraction, or in this case it will be called like the pumping, such kind of pumping, muscle pumping system will be responsible for venous return of blood. Venules, it is the smallest part of the veins. They have thinner walls in comparison with arteries. Post-capillary venules, it is the type of the smallest venules to which will blood flow through the capillaries, and they are also the part of the microcirculatory exchange, which was unit with the capillaries. Muscle venules have a thicker walls with 1 or 2 layers of the smooth muscles. So in this picture, you could see the structure of these valves, which are present in the vein system, and these valves are responsible to prevent retrograde flow of blood to the capillary system. And the main function, as I mentioned previously, is very important in case of the veins which are located in the limbs, because in this situation, muscle pump or contraction of the skeletal muscle will be responsible to pumping the blood, to help to move blood through the vein system and through these veins toward to the heart. Mainly when we talk about the circulatory blood distribution, the venous system, it is the blood reservoirs, which reserves about 64% of the blood. It's a large portion of the blood will be stay at the rest here. It's happened due to the low pressure which is present in the venous system. For instance, in a venous system, the pressure could vary from the 10 millimeters per mercury. For instance, in the vena cava superinferior, it's about 4 to 5 millimeters per mercury, so the pressure is here very low. But due to the influence of the sympathetic division, it could lead to the vasoconstriction of the vessels of the venous, and this will lead to increased blood pressure in this system in case when it is needed. Okay. Then we are going to continue to talk about the basic concept of hemodynamics, blood flow, resistance of blood flow, and blood pressure. Now we are going to discuss the second part of our lecture. It is about basic concept of the hemodynamics, which will be presented by the blood flow, resistance of the blood flow, and the blood pressure. Mainly the principles of the blood circulation will be discussed on the level in the vessel will be called the hemodynamics laws. So now we are going to stop on the different kinds of the hemodynamics laws. First of all, let us discuss the blood flow. So what is the blood flow? What is its concept? It means the quantity of blood that passes a given point in the circulation in a given period of time. Mainly, the overall blood flow in a systemic circulation is identical to the cardiac output. So when we talk about blood flow, we could mainly mean the cardiac output. So what factors will be responsible to determining of the blood flow? The first factor will be interrelationship among blood flow, pressure, and resistance. So when we wanted to identify what will be the value of cardiac output, we first of all need to interesting on the differences of pressure at the beginning and at the end of the vessels. When we talk about the differences of pressure at the beginning and end of the vessels, we're interested in the mean pressure at the beginning, which will be presented for us mean arterial pressure, and at the end of the vascular system, we are going to talk about the mean venous pressure. So the differences between mean arterial pressure and mean venous pressure is one of the important things, which will determine our blood flow. Another factor which is going to determine our blood flow, it will be friction force produced when blood flows through the blood vessels. In other words, it is the resistance of the vessels. So Q, or blood flow, or we could name it like cardiac output, is equal to, it will be directly proportional to the differences of pressure, as we identify differences of pressure of mean arterial pressure with mean venous pressure, and indirectly proportional to the resistance of the vessels. So pressure gradient, you could see here that at the beginning of the vessel, the pressure could be about 50 millimeters per mercury, and at the end of the blood vessels, the pressure is going to be 10 millimeters per mercury. So the differences pressure in this vessel 1, it will be 40 millimeters per mercury. But if we compare, so you could see how blood will flow in this type of vessel. But if we compare vessel 1 with vessel 2, you will see that in the vessel 2, we have a higher pressure at the beginning of the vessel, 90, and the same pressure at the end of the vessels, 10 millimeters per mercury. So in this vessel, the differences of pressure will be 80 millimeters per mercury, and you could compare the different type of blood flow. The blood flow in the second vessel will be bigger, higher, and the volume will be bigger in comparison with the first one. The same we could see in the pressure, in the vessel 3, where the blood pressure will be at the beginning of the vessel will be about 180 millimeters per mercury, and at the end of the vessel it will be 100 millimeters per mercury. But nevertheless, the differences between these two pressures will be 80 millimeters per mercury. So the blood flow will be the same as in the vessel number 2. So pressure difference, say it's mainly effect on the blood flow, not absolute pressure, but the pressure difference is effect on the blood flow. Another point which is going to affect on the blood flow, it is the resistance of the blood flow. In this situation, we are going to use the Poisson equation, which means that resistance will be directly proportional to the viscosity of blood and the length of the blood vessels, and indirectly proportional to the radius of the blood vessels. If radius, please look here, if radius decreases by 1/2, resistance in the vessels will increase by 16 volts. So in this case, we could say that the main factor which is responsible for determination of the resistance of the blood flow will be the diameter of the vessel. Now let's discuss this moment in detail. So radius profoundly affect blood flow. So in vessel 1, for instance, the blood will flow with the speed which is about 1 millimeters per second. But when the same vessel, the diameter of a vessel is going to increase twicely, the speed in this vessel will be bigger. So from 1 millimeters per second, it will increase on 16 volts, so it will be 16 millimeters per second. So generally, the velocity of the blood flow in the vessel number 2 is going to increase. You could see it even in this picture presented. Same pressure gradient, but the different resistance, and as a result, the different speed or velocity of the blood flow. The velocity of blood flow mainly will depend on, mainly will depend on the velocity will depend on the flow, which is measured by millimeters per second, which is going to be divided on the cross-sectional area. So when the cross-sectional area will become, you could see here, yes, that the area of the cross-sectional area of the small vessels is about 1 centimeter in quadrat. In middle vessels it will be 10 centimeters, and in big vessels it will be 100 centimeters. So generally, the flow, you could say that the velocity will change. Bigger cross-sectional area, so velocity will become bigger in case when the cross-sectional area is going to be smaller, and vice versa. So velocity of blood flow, its speed in centimeters in a second is inversely related to the cross-sectional area. Velocity is the slowest where the total cross-sectional area is the greatest. When we have a greatest cross-sectional area, velocity of the blood flow will be smaller, and vice versa. Velocity will be bigger in case when the cross-sectional area is going to be small. Blood flow becomes slower further from the heart. So it will decreases when the blood flow out from the heart. The slowest blood flow is going to be in the capillaries. And we know that the main function in capillaries is responsible for the exchanges. Circulation time, it's time required for a drop of blood to pass from the right atrium through the pulmonary and systemic circulation and back to the right atrium. Normally it will duration 1 minute at rest. So in this slide, we have presented the relation between velocity of blood flow and the total cross-sectional area in a different type of blood vessels. So we could see that cross-sectional area in aorta, arteries, aorta and artery will be small, but besides this, the velocity of blood flow will be higher. But cross-sectional area is going to increase on the level of the, to some extent, arterioles, but then it will be very high on the level of the capillaries. And in this level of the capillaries, you could see that the velocity of blood flow with the greater cross-sectional area will be smaller. And if we continue, we could see the cross-sectional area will become smaller in the case of the veins and the vena and the vena cava system, and velocity less slightly, but nevertheless it will try to increase. So the conclusion we could make like this, that if we have higher velocity, high velocity mainly present in vessels with a small cross-sectional area, it means that vessels which have a bigger diameter. And in vessels which have a smaller diameter, they will have a small, they will have a greatest cross-sectional area, and logically the velocity of blood flow in this region will be slower. Also, when we talk about the blood flow, we could divide it that blood flow could be different type, could be laminar type or could be turbulent type. So here is presented laminar type. Laminar flows is parabolic, highest velocity is in the center, lowest adjusted to the vessel walls. You could see here that the highest velocity will be presented in the center of the vessel. The slowest velocity will be presented near the walls of the vessel. That is why in such vessels, erythrocyte mainly will be concentrated in the center of this blood flow, and near the walls will be concentrated mainly plasma, which will, as a result, will receive some kind of friction between the vessels and the plasma. And the turbulent flow is disorientative, no longer parabolic, energy wasted, thus more pressure required to drive blood flow. So in this situation, you could see it will be like a noisy. There is nothing will be arranged in this kind of the turbulence. Sometimes laminar blood flow could convert it to the turbulent. This could be happen due to the differences, due to the changes of the viscosity of the blood, due to the changes of its diameter. So mainly in this picture, we could repeat once more that arteries, that the vessels which that in the laminar blood flow, the high velocity will be in the center, and sometimes sometimes this laminar will convert it to the turbulent, but then it will relate but further it's also able to convert to the laminar blood flow. Laminar blood flow is more greater in the vessels with a bigger diameter. In this situation, we have more place for pumping blood through the vessel system. And in case when we talk about when the diameter of the vessel will become smaller, this will lead to, this could cause to change the type of blood flow to the turbulent. And in vessels with a small diameter, mainly the blood flow is going to be smaller. This has happened because the center part will be decreased, and which is mainly responsible for the high velocity. And as a result, red blood cells will are more vulnerable to connect with the walls of the vessels, and as a result to aggregate. This will lead to some extent to the stop of the blood flow to produces of aggregates of the red blood cells, platelets, and further to the thrombosis. Okay, so we could say the turbulence could happen due to the changes of the diameter of the blood vessels. And also another factor which could affect on the conversion laminar type to the turbulent, it is a change of the viscosity. When the blood viscosity will increase, it will also be responsible to decrease speed of the speed of blood flow in the vessels, and mainly it will be more characterized in vessels with a small diameter like arteriolus or capillaries of the veins. And this situation could lead to the turbulent, to the conversion laminar type of blood flow to the turbulent one. So also there is another type of blood flow. It's like a plug flow, when all molecules move at the same speed. Happens only at very small diameters vessels, and the slow and the velocity is going to be very slow. Laminar flow due to the friction against vessel walls. The blood near the center of the tube flows faster than on the periphery, as we discussed. Infinitesimally, the concentration cylindric sliding past each other. The velocity profile is shaped like a parabola. And the turbulent flow, or a chaotic random, occurs when the Reynolds number for a fluid is exceeded. So the next point which you're going to talk, it will be about blood pressure. Now let us discuss about blood pressure. So what is the blood pressure? It is the force exerted by the blood against the vessel wall. Mainly it is the main pressure which will be forming as a systemic vascular circulation. Also it's responsible to some to store energy for potential energy. When we talk about blood pressure, we know that it is arterial pressure will be divided in the systolic pressure, which happens, it describes the function of the heart during the systole. Diastolic pressure, which describes function of the heart in period of the diastole. Mean pressure, it is average value of the systolic and diastolic pressure, and pulse pressure, which describes the differences between systolic and the diastolic pressure. How we can count mean arterial pressure? It will be diastolic pressure, which should plus 1/3 of the pulse pressure. Everything that happened at the rest. So this time mainly will take part in the diastole, and it's in a time it will be presented by 40 minutes. It will be 80 millimeters per mercury of the diastolic pressure, and once a time is mainly presented by the systole, is described by the 20 minutes, and it is 120 millimeters per mercury pressure in the period during the systole. So what does systolic pressure tell us? Systolic pressure mainly tells us information about the pressure in the large arteries, like aorta, which happens during the systole of the heart, during the heartbeats. And what does diastolic pressure tell us? It tells us information about the pressure in the big, large arteries. During the diastole, it means between heartbeats, during the period when the heart is in the rest. Mean pressure, you could see here, mainly it is described pressure which will be present in the arterioles. In upper, you could see here that the cardiac output, so the blood flow which flows through the whole circulatory system depends on the mean arterial pressure, and also directly, depends on the mean arterial pressure, and indirectly depends to the tissue peripheral resistance. About these values we are going to discuss a little bit later what they mean. How aortic pressure could change. So generally, when we have when we know that the aorta is related to the type of the arteries which have a very developed elastic fibers. Due to this, they could, without any problems, dilatate due to the big amount of volume which pumps through the left ventricle to the aorta, and as a result, it will produce the pressure. So it is a normal example how the waves of the blood flow which will be presented in the aorta it will be presented by the pressure. Sometimes in the aorta there is such kind of diseases like atherosclerosis, which could lead to the rigidity of the blood vessel. It means it will be difficult for aorta to change its shape, And as a result, in this atherosclerosis, it's one of the important factors which is going to lead to the atrial hypertension. So during the atherosclerosis, you could see that the pressure in the aorta will become very high, very big. It's happened due to the rigidity. And in case when we have the stenosis of the semilunar valves or aortic valves, the pressure in aorta will be very low, because in this situation it will be rather difficult for left ventricle to pump blood through the stiffness, through the narrowed valves, and as a result, small volume and small blood flow will deliver to the aorta. So in this picture, we could see how normal is that this is a container of the water. It's like example of your heart, which is full of the blood. And when its valve is open, we could see that the blood will flow through the rubber tube or the glass tube normally with a steady pressure. But in case if we open the valve and then rapidly close it, you will identify that the blood flow in aorta, in our case it will be presented by the tubes, will be different. First of all, big amount of volume which pump to the aorta will lead to the increased blood pressure, and due to its, and after that it will lead to and after this situation will happen, due to the closing of the valve will happen the decreased volume in aorta. But nevertheless, that outflow will be such rather big with increased pressure if it's happened due to the rigidity. In case of the pulsating rubber tube, yes, due to the high volume which will be pumped to the aorta, aorta is going to change its shape, it means dilate. But when due to the quick decrease of the blood flow, according to the stenosis, it will return to its previous shape. So we will have such kind of pulsation in the situation of the rubber tube. In our case, it will be pulsation or changing of the diameter of the aorta. And you will see that the blood flow will be it's similar to like expressed here for aortic stenosis, and the pressure will be lower. Sometimes, in case of the aortic regurgitation, if we compare aortic stenosis with aortic regurgitation, due to the insufficiency of the aortic valves, blood will flow back during the blood will flow through these insufficient valves to the aorta. So the volume in the aorta will also increase. Mainly when we talk about the blood flow and when we talk about the pressure, first of all, we need to we discussed that the cardiac output depends on the mean arterial pressure, but also it depends on the tissue peripheral resistance. We talk about the tissue peripheral resistance, it mainly depends on the small vessels, small arterioles, capillaries, venules, which is the part of the which have the higher cross-sectional area. So the capillary exchange, which is a part of this high cross-sectional area, also will play a role in the change of the arterial pressure. In the capillary, this will happen due to the precapillary sphincter which are present, which could be opened and responsible to vasodilatation and decrease systemic blood pressure, or in case when we have high systemic, in case when we have the low systemic blood pressure, the precapillary sphincter are able to close, and as a result try to increase the systemic blood pressure. So let us discuss about the capillary exchanges. Capillary exchanges, movement of substances which happen between blood and intestinal fluid. There are the three basic methods of the capillary exchange. It is a diffusion, transcytosis, and black flow. What is a diffusion? Diffusion, the most important methods. Substances move down their concentration gradient, so from the high concentration to the lower concentration. Oxygen and nutrition from the blood will move to the intestinal fluid to the body cells. Carbon dioxide and wastes move from body cells to intestinal fluid and also to the blood. During the diffusion, the oxygen and carbon dioxide could cross capillaries walls through intracellular cleft, fenestroids, or through the endothelial cells. Most plasma proteins cannot cross this, except in case when we talk about the continuous capillaries. In case when we talk about the sinusoid capillaries, proteins and even some blood cells could leave the vessels and transport diffuse to the interstitial fluid, intracellular space. And the example of such station will be blood-brain barrier, tight junction with a limit diffusion. Transcytosis, it happens during the small quantity of material. Substances in the blood plasma become enclosed within the penocytotic vesicles that enter endothelial cells by endocytosis and leave by exocytosis. So mainly by the transcytosis, large moleculars, lipid-insoluble moleculars can be enclosed in a penocytocytic vesicles, and then by endothelial cells they will leave vessels through the process which is called exocytosis. And blood flow. It is a passive process in which a large number of ions, molecules, or particles in a fluid move together in the same direction. Mostly it is based on the pressure gradient. Diffusion is more important for solutic change. Blood flow more important for regulation of relative volumes of blood and intestinal fluid. Filtration mostly will happen from the capillaries into the interstitial fluid, and reabsorption will happen backward from the interstitial fluid back to the capillaries. To discuss about this filtration reabsorption network, we need the equation which is present here, which is called net filtration pressure, which mainly based on the balance of the two pressure. So the first pressure, it will be blood hydrostatic pressure, which could be different. For instance, at the beginning it could be 35 millimeters per mercury. At the end it could be about 15 millimeters per mercury. Then we should sum it up to the interstitial fluid osmotic pressure, which the average value is 4 millimeters per mercury. Then we need blood colloid osmotic pressure. Mostly the blood colloid osmotic pressure is based on the level of the plasma proteins, and its average value is 25 millimeters per mercury. And interstitial fluid hydrostatic pressure, which is generally about 3 millimeters per mercury. So if we put these values on this net filtration network, we are going to receive such a result that on the artery part, on the beginning of the process of filtration, it means on the precapillary, the arteriolus part, the net filtration network is going to be 11 millimeters per mercury, and at the venous end, the net filtration network is going to be minus 9 millimeters per mercury. So due to these differences, we could say that about 85% of fluid which is filtrated will be reabsorbed. So mostly all fluid will be filtrated, but due to the low level of the net filtration network, net filtration pressure in the venous end, only 85% will be reabsorbed back. This another percentages, within percentages will be transported by the lymphatic capillaries. Mainly it's about 3 liters a day, and then they eventually will be returned back to the blood. So what could affect on this net filtration pressure? Increased level of the hydrostatic, blood hydrostatic pressure at the beginning on the arterial end could lead to increased process of filtration, or to some, as a result, in case when we have another situation, when we have decreased level of colloid osmotic pressure in the venous end, it will lead to the decrease of reabsorption, and as a result, it will lead to the edema condition. So in this picture, we could see the dynamics of the capillary exchange. We have the hydrostatic pressure inside of the vessel will be 35 millimeters per mercury. You could see that the blood colloid osmotic pressure, it will be equal as at the artery end as in the venous end, but the blood hydrostatic pressure in the venous end will be smaller in comparison with the artery. It will be about 16 millimeters per mercury. And you could see how, if we put all these values in our net filtration equation, so we will see in the artery end it will be about 10 millimeters per mercury and -9 in the venous end. So 3 liters of blood flow will be transferred through the lymphatic system, and then they will return back to the vascular system. So now let us, to some extent, let's return back to the blood pressure and its control. So mainly contraction of ventricles will play main role for generation of the blood pressure, for generation of the systolic blood pressure, and for generation of the diastolic blood pressure. Systolic blood pressure, it is the highest pressure which is happened in the arteries during the systole, and diastolic blood pressure is the lowest arteries pressure during the diastole of the heart. Pressure falls progressively with the distance from the left ventricle. Also, we should mention that the blood pressure depends on the total volume of the blood. So when the blood volume will decrease, logically that the blood pressure, that the situation will lead to decrease of the blood pressure. Normal range of the arterial pressure. Systolic, as we talked, pressure is the maximum pressure during the systole. Its normal value from 120 to 139 or 40 millimeters per mercury. Diastolic blood pressure is the minimum pressure during the diastole. It's about from 60 till 80 millimeters per mercury. Pulse pressure is the differences between systolic and diastolic pressure, and the mean arterial pressure is the average pressure through each cardiac cycle. How we could count mean arterial pressure? We need diastolic pressure plus pulse pressure divided it on the 3. So this is the equation for receiving the general value of mean arterial pressure. How we, in this slide, you could see the graph which describes this value, that the systolic pressure, it's a red line, is rather higher, diastolic pressure should be lower, and between them should be located this mean arterial pressure. It shouldn't be higher than systolic and shouldn't be lower than diastolic one. How we could measure arterial pressure? Mainly we use nowadays, it is indirect auscultation method or carrot cough method. We use such specific devices like stethoscope and sphygmomanometer. Here's in this picture presented how the doctor measures pressure in the patient. What happens during this situation? When you put a cuff in the arm, first of all. Secondly, we need to pump pump the cuff, and in this case there will be increased pressure and it will restrict the vessels on the artery. And when you pump it, when the cuff pressure will be more than 120 millimeters per mercury, you could see that we make the occlusion of the arteries, so you will not feel, so you will not hear any kind of sound from the stethoscope. When cuff pressure, when you will switch off the calf, and as a result the pressure, the air will go out from it, you are going to start to listen the sound. The first sound which you're going to hear, it will be the first sound which will be the first critical sound, which will be created by the pulsated blood flow through the compensated artery, and this will be the systolic pressure. First, this systolic pressure will be not very loudly. Its loudness is going to increase, and the last sound which you are going to hear, it will be the the diastolic sound. It will describe, I'm sorry, the diastolic blood pressure, and after that you will no longer hear any kind of sounds. So, basis of auscultation method for measuring of blood pressure. So, how sounds of correct cough will appear. So, you could see that when the cough pressure will be more than 120, mainly it will be, you should pump it to 180. As a result, you will not hear any kind of sounds from the stethoscope. But then no sound, you see. But then, when you will switch off that cough, the air will go out, and you and the vessel will not be constricted by that cough. So logically, you are going to start to listen the sounds, which is responsible for systolic. The first sound will be responsible for systolic arterial pressure. You could see here that it will not be very loud, but it will further, it's going to increase and become very loud and soft, which will mainly, and the last sound which you're going to hear, it will be will describe the diastolic blood pressure, and after that you will not hear any kind of sound. It means that the blood pressure returned to its normal value. Vascular resistance, we also talk about that the cardiac output depends not only to the mean arterial pressure, but it also depends on the vascular resistance. Vascular resistance depends on the size of the lumen. Vasoconstriction, males lumen smaller, meaning greater resistance. Blood viscosity, it means rate of double red blood cells to plasma and protein concentration. High viscosity means higher resistance. It means total blood vessel length also affect on the resistance directly. But mainly the main factors which affect on the resistance, it will be the size of the lumen, it is vasoconstricted or dilated vessels, and also depends on the viscosity. In case we have the situation of the vasoconstriction, it will lead to the increased tissue peripheral resistance. Venous return. A venous return is in another name of it, we could name like afterload. So blood pressure also will depend on the level of the afterload. When the venous return will be high or big, so as a result we have the extension of the atriums and ventricles of the heart, and logically that the stroke volume of cardiac output, which will be pumped through the heart, will be bigger. And increase of blood flow or cardiac output logically will lead to increase of blood pressure. So venous return, it's a volume of blood flowing back to the heart through the systemic veins. It's occurred due to the pressure generated by constriction of the left ventricle, and also it's happened due to the differences of pressure between vena cava superior inferior and the right ventricle. In the vena cava superior inferior, it could be about 4 or 5 millimeters per mercury. In the right ventricle, first of all, it will be 0 millimeters per mercury. So due to the differences of pressure, blood will flow towards to the right atrium. What helps is, in case when we discuss, talk about the vena cava superior inferior, but before we need to also stop on the venous, on the veins which are responsible to deliver blood from the lower part of the body, from the legs, and it will happen due to the anti-gravity system. So what mechanism will help to pump the blood towards to this vena cava superior, vena cava inferior, I'm sorry. The first mechanism, it will be skeletal muscle pump. Muscles and another mechanism, it will be respiratory muscle pump. In case of the skeletal muscle pump, we could see in this picture, you see that the muscles of the calf, they, due to the movement, due to the, for instance, even walking, they will contract, and due to their contraction, the distant wall will close, and as a result, and the proximal veil will open. So it will allow blood flow toward to the heart, to the vena cava inferior, and prevented blood flow back. And also, as a result, in this situation, it will help to increase venous return to the heart. In case when we talk about the respiratory pump, it's happened due to the thoracic cavity. Mainly during the inhale, the pressure in the thoracic cavity will decrease, but at the same time, the diaphragm will be pulled down and the pressure in the abdominal cavity will become higher. And this will help the blood also pump upward to the heart. Okay, so when we look through this graph, slide, I'm sorry, we could see what factors were responsible to increase mean arterial pressure. If responsible to increase mean arterial pressure, it will lead to increase of the generally of the systemic blood pressure. So increase of blood volume, logically, which will be in the circulatory system. Due to the circulation, this increase of blood volume will return back to the heart through the function of the skeletal muscle pump, respiratory muscle pump. Also due to the effect of sympathetic division on the vessels, which will be responsible for vasoconstriction. So all of these factors will be responsible to increase venous return. Increased venous return, first of all, will be responsible to increase stroke volume and as a result, increase of heart rate, cardiac output. So this is the first line of mechanism how we could increase mean arterial pressure. Another situation, depression of parasympathetic impulses on the heart, yes? So depression of the parasympathetic impulses and increased sympathetic impulses will be responsible to increase heart rate. Also, we know that sympathetic division will be responsible not only to increase the heart rate, but also responsible to increase stroke volume, which together will increase cardiac output, which will lead to increase of the mean arterial pressure. Another mechanism how we can increase mean arterial pressure, not only through the function of the heart, but also through the systemic vascular resistance. This could happen due to the systemic vascular resistance could increase in case when we have high blood viscosity. This situation could happen in case when we have a ratio of red blood cells bigger than the plasma, or so it will be such conditions like polycythemia. Also, increased total blood vessel length. Mainly this happens in case of the obesity, because when the person during the obesity person increases its sizes, so adipose tissue requires more delivery of oxygen, new process of neurovascularization. Everything of this will lead to the increased total blood vessel length and also their cross-sectional area. And as a result, another mechanism is vasoconstriction, which could happen also due to the effect of sympathetic division. So these three mechanisms were responsible to increase systemic vascular resistance, which then directly will impact to increase of mean arterial pressure. What is the main center which is also responsible to regulate blood pressure? It is the Cardiovascular Center, which is located in the medulla oblongata. This center is responsible for regulation of the heart rate and the stroke volume. It is responsible to increase the heart rate or stroke volume or vice versa to inhibit it. Also, it controls neuronal, hormonal, and local negative feedback system. In the cardiovascular center, we have cardiostimulatory and cardioinhibitory center. center, and also we have osmotic center, which will control blood vessels diameter. Information to the cardiovascular center will be transmitted through the sensory information from the peripheral baroreceptors of the chemoreceptors, which are mainly located in the aorta or in the carotid sinus. So, cardiovascular center, you could see here that it will input information, first of all, from the higher brain center, like cerebral cortex, limbic system, or hypothalamus. In this situation, they will affect on the vasomotor center, which is responsible for increased is responsible for constriction of vessel, or responsible for effect on the heart and increased heart rate and its contractility. How cerebral cortex, limbic system, or hypothalamus could affect on this cardiovascular center? So, for instance, limbic system or hypothalamus, they could affect due to some kind of huge weak emotions, which could happen, for example, during the stress. The same, some kind of stress conditions, your consciousness stress conditions, some kind of your during the exams or something like this, when you will be very nervous, the same situation, cerebral cortex will play part in the influence on this cardiac accelerator center or vasomotor centers. Also, information could be sent through the monitor for the joint movements. So during the physical activity, it means, yes, logical, that during physical activity, the heart rate and its contractility and blood pressure increases. And through the baroreceptors and chemoreceptors, which are located in the aorta and the carotid sinus, due to the increase of the blood pressure, and which will be responsible to increase size or diameter of the aorta or the carotid sinus. This increase of diameter of the vessels will be detected by the baroreceptors, send it up to the cardiovascular center, and responsible for inhibition of the vasomotor and accelerator center. As a result, prevent further constrictions of vessel and increase the heart rate and its contractility. So, three main types of sensory receptors. Proprioceptors, as I told, they monitor movements of joints and muscle to provide input during physical activity. So, logical, during physical activity, heart need to contract with more frequently and with a big force in case to pump more blood, which is further going to deliver to all tissues and organs in the body. Baroreceptors, they mainly monitor pressure in the aorta and carotid sinus, the ability to stretch or expand due to the increased blood volume, or stretch due to the decreased blood volume in the vessels. Chemoreceptor will monitor the level of the oxygen and carbon dioxide in the blood. So when we have the condition of the hypoxia and hypercapnia, which means decreased level of oxygen and increased level of carbon dioxide, this will lead to the this will be detected by the chemoreceptors in aorta and carotid sinus and transport upward to the medulla oblongata, and in fact in two centers. The first center is cardiovascular center. As a result, we will receive increased heart rate and its contractility. And besides this, it will affect on the respiratory center, which will be responsible to increase the frequency of breathing. It means to increase level of oxygen in the blood and remove huge amount of carbon dioxide, which lead to the hypercapnia. Also, systemic, so sympathetic and parasympathetic division, they affect directly, for instance, to some extent on the heart. So they're responsible, for instance, parasympathetic division to decrease heart rate and its contractility. As a result, it will lead to decreased blood pressure. And sympathetic division also affect on the vessels, vascular system, and it will be responsible not only to increase heart rate, but also to increase vasoconstriction. So generally, the neural regulation, the baroreceptor reflex, as I explained you, it's a pressure-sensitive receptors in the internal carotid arteries and other large arteries in the neck of the chest. chest, which are going to detect the volume which will be present in this system, or it will be increase or decrease. So the arteries will or stretched or dilatate. So when blood pressure falls, baroreceptors stretched. Less, slower rates of impulses to cardiovascular center. Decreases cardiovascular center, decreases parasympathetic stimulation and increase sympathetic stimulation. Chemoreceptors. Receptors also located near the baroreceptors in the carotid sinus or aortic arc. They detect hypoxia and hypercapnia, condition of the acidosis. Cardiovascular center increases sympathetic stimulation on arterioles and veins, producing vasoconstriction and an increase of blood pressure. Besides this, receptors also provide input to the respiratory centers to adjust breathing rate. So mainly here, everything was the same information we'll describe here, of sympathetic division and effect on the heart, and how the information from the baroreceptors of the aorta and carotid sinus will be transmitted afferent information to the cardiovascular center through the spinal cord. Also, function baroreceptors, chemoreceptors, they're responsible for short-term regulation of blood pressure. But besides this, we have a long-term regulation of blood pressure. It means that this is a system which is going to regulate blood pressure for some period of time longer. Example of the long-term regulation of blood pressure, it is the renin-angiotensin-aldosterone system. Renin mainly will be released by the kidney when blood volume falls or blood flow decreases. And angiotensin converting enzyme is a substance like enzyme which is responsible to convert angiotensin on angiotensin 2. It's very important. Another the subhormone which is responsible for vasoconstriction, first of all, which will lead to increased blood pressure. But besides this, it will affect on production of releasing of aldosterone by adrenal glands, and as a result, aldosterone is responsible for reabsorption of sodium, and besides this, indirectly responsible for reabsorption of water from the kidney. And in this situation, we have increase of blood pressure not only due to the vasoconstriction but also due to the return of the blood volume. Epinephrine and norepinephrine, will be released by the adrenal medulla in response to the sympathetic stimulation in a stress situation. And also they have the same effect as sympathetic division, increase heart rate and vasoconstriction. Antidiuretic hormone or vasopressin, which is produced by the hypothalamus and released by the posterior pituitary gland. This hormone will be released due to the dehydration and decreased blood volume. It means that this hormone will directly affect on the kidney, and it will responsible for direct reabsorption of water. As a result, we'll return back water to the vascular system. In one way, we are going to increase blood pressure, but besides this, ADH also have effect on the vessels, and it will lead also to the vasoconstriction. Atrionatriuretic peptide, it have the vice versa effect to the antidiuretic peptide. It will be released by the cardiomyocytes of the right atriums, and its mechanism is responsible to decrease blood pressure. When we have due to the big venous return, we have the extent of the cardiomyocytes of the heart. Due to the most, we have the extendance of the atriums, of the right atrium. As a result, ANP is going to produce, and it will affect directly on the kidney, and it will be responsible to increase excretion of the water excretion of the water by the kidney, so increase the urination. As a result, blood volume will decreases, and further venous return will become smaller. So this arteriolus peptide have two mechanisms. The first mechanism is responsible to decrease blood volume, and the second mechanism is to prevent the overextension of the heart, and further to prevent its it has a protective mechanism for the dilatation of the chambers of the heart. Another mechanism, it is auto-regulation of the blood pressure. It's due to the ability of the blood vessels, mostly we talk about the arterioles, to change their diameter due to the metabolic demands. And there are two general types of stimuli which could affect on it. It could be physical activity, factors like temperature changes, myogenic response, and vasodilating vasoconstricting chemicals like nitric oxide, carbon dioxide, responsible for vasodilatation of the arterioles, but endothelin or angiotensin 2 is responsible for its vasoconstriction. Generally, thank you for your attention. Lecture is over.